let us look at the topic biotransformation or microbial transformation. So biotransformation is a biological process where one organic compound is converted into a chemically similar organic compound. Okay, so these are catalyzed by one or two reactions which are very simple reactions using the enzymes present inside the cell. The cell can be plant cell, animal cell or microbial cell, but in our case we are more interested in the microbial transformation, microbial biotransformation, or it can be also referred to as microbial transformation. Okay, so what is microbial transformation? When the biotransformation or the transformation of organic compound is carried out using a microorganism, this is a microbial transformation. So what is the difference between a microbial transformation and a fermentation process? In the fermentation process also the microorganism is carrying out a conversion, but here the difference is the microorganism is converting one form to another, and in the biotransformation process only a few steps will be there, a few steps of conversion will be there. But in a fermentation process it is not a direct conversion into related structure, and multiple steps will be there. Like from the carbon source will be the sole used. The carbon is converted into something else, it's again converted. So multi-step process is happening in a fermentation. Okay, so how is the microorganism transforming an organic compound? The microorganism will be having so many enzymes. Okay, and these enzymes will act upon the biological molecule or the organic compound, and it will convert it to another compound. The most oldest example is the production of vinegar, where wine is converted into, or alcohol is converted into acetic acid. Okay, alcohol to acid conversion. So when we are comparing microbial transformation with the plant and animal transformation, so we can also use plant cells and animal cells for transformation, because bio means you are using a living cell. But when we are comparing microbial transformation to the other two methods, microbial transformation have so many advantages. So looking at the advantages, microorganism has high surface to volume ratio because of the small size of microorganism, and this high surface to volume ratio enable the efficient conversion of the organic compound. Okay, the growth rate of the microorganism is very high compared to the plants and animals, so it is less time consuming. You can easily maintain the sterility of the microorganism. Okay, but in case of plants and animal it is very difficult. It is easily contaminated, the animal cell culture and the plant tissue cultures. So the metabolism rate of the microorganism is also very high compared to the plant and animal culture. So microorganism is preferred over the other, microbial transformation is preferred over the other types of transformation. So apart from biotransformation, initially what was happening is, when you want to produce one organic compound, okay, for example you want to produce a steroid tablet, so you will be using a chemical starting material and you will be converting into whatever you need, a steroid tablet or a steroid substance via chemical reaction. So these chemical reactions, there will be multiple chemical reactions happening, and these will be like having like so many input also should be there. So it is economically draining, so that whatever is produced will be very costly, and since multiple reactions are there, it will be time consuming also. So let's also compare the microbial transformation with chemical synthesis. So microbial transformation, in case of microbial transformation, microorganism has a enzyme which is acting upon the organic compound. So there is a high substrate specificity. Apart from the substrate specificity, there is also type specificity. Okay, there will be several functional groups that is present on an organic molecule, but when you are doing biotransformation, none of the other functional groups are affected and only specific position may be converted. Okay, example is progesterone. It has several functional groups, and by using Rhizopus nigricans you can convert progesterone to 11 alpha hydroxy progesterone. At a single position there is a hydroxy group being added. Then the enzymes are also showing stereoselectivity. If you are having a racemic mixture, the enzyme will act on a specific enantiomers and convert it. Okay, so this is also, the specificity is very high in case of microbial transformation, and the number of steps and the economic input is also very high in case of chemical transformation, chemical synthesis. So microbial transformation is again preferred over chemical synthesis. So when we are looking at the microbial transformation or bioconversion, there are different categories of chemical reactions that can occur, like oxidation, reduction, hydrolysis, condensation, isomerization, etc. Okay, and oxidation reaction is the most useful one in industrial production, and to a lesser extent isomerization, reduction, hydrolysis and condensation are also applied. So it's not always that a single reaction is carried out, just an oxidation. No, more than one type of reaction can be carried out in a biotransformation. Okay, one or more than one, and the time required for a biotransformation to complete is depending on the type of reaction that is happening, the substrate concentration and the microorganism that is used. And if the type of reaction is oxidation, hydrolysis or dehydration, then it is completed very, very soon. So how do you get the biocatalyst for, like, what are the biocatalysts that can be used for biotransformation? You can use growing cells, resting cells, you can use killed microorganism, immobilized cell, immobilized enzyme, free enzyme, cell-free extract. All these can be used for converting or for biotransformation or bioconversion. Okay. So when you are using non-growing cells, these are preferred over growing cells. Okay, why? Because when you are using a high concentration of substrate which has to be converted or which has to be transformed, normally what happens is, when you are using a growing microorganism, the growth of the microorganism stops due to the high concentration of the substrate. But when you are using the non-growing cells, very high concentration of substrate can be converted. Okay, then non-growing cells can be easily washed and used, so the contamination can be reduced. The conversion efficiency is very high in case of non-growing cells. That means the cells that are not in the growing stage. Okay, and the production, the isolation and the recovery is easy. There is easy optimization of the condition. Okay, then you have immobilized cell. Okay, you can also use immobilized cell. Okay, so you can reuse the cells for the biotransformation reaction. Okay, biotransformation which are carried out in a single or a multi-stage reaction can be done by immobilized cell. For example, L-alanine and malic acid production have been carried out using immobilized microorganism. You can also use immobilized enzyme, cell-free enzymes for biotransformation, because when you are using immobilized enzyme, there is no unwanted side reaction. That is, there is only a single enzyme or one or two enzymes that are specifically needed, so you don't have any other reaction that are occurring, and the desired products are not degraded, because there are certain enzymes that will be present in the microorganism which will carry out further degradation or further metabolism of the product whichever is being formed. But when you are using an immobilized enzyme, you can only conduct those reactions that are necessary for you. There is no transport barrier across the cell membrane. You have a free enzyme that has been immobilized, so transport across the cell membrane is not restricted, and hence the isolation recovery is very, very easy. These are two examples where immobilized enzymes have been used. So after the product have been formed in biotransformation, the recovery is generally easy because mostly the products are extracellular. Okay, it will either be in a soluble or a suspended form. Okay, and when you are using a cell for biotransformation, it has to be separated using water or organic solvent depending on what is your product. Okay, is it water soluble? Is it a non-polar? Depending on that, you will be separating it after the production. Okay, then you can use the normal downstream processing like precipitation by salt, extraction with solvent, ion exchange, chromatography, etc. And the volatile, if you are going for a volatile substance, it can be easily distilled. So let us look at a few examples of biotransformation. One of the main example you can know is production of acetic acid. That is a very good example. Okay, another one is biotransformation of steroids. Okay, steroids are natural products, okay, that are widely distributed in nature. Steroids are present in sex hormone, it are present in alkaloids, some antibiotics are steroid containing antibiotics are there. Okay, so many steroids are there. Okay, so some examples are Corynebacterium species have been used for the biotransformation of dihydroepiandrosterone into testosterone. Okay, and Nocardia species have been used to produce cholesterol from 4-dehydroethocolanic and 7-hydroxycholesterol. From these two, the cholesterol have been produced using the organism Nocardia. Okay, now looking into the steroid production, steroids have a similar basic structure. Okay, and this is having a 4-membered fused ring structure. So the mostly used steroids are cortisone and prednisolone because it is anti-inflammatory and it is used for the treatment of rheumatoid arthritis, skin diseases, etc. And these are produced using the organism Arthrobacter, okay, by a biotransformation reaction, and cortisol can be transferred into what? The other one by biotransformation reaction. Then looking at the steps for production of steroids, initially steroids were produced by just chemical transformation. But as discussed, chemical transformation is involving so many steps and so much of money has to be inputted, and hence the price of the tablet or whatever will be very high. So now chemical transformation in combination with microbial transformation is generally preferred. And usually the production of steroids is done as a batch fermentation and immobilizers or enzymes are used. Okay, why? Because these can give a very short conversion time. Steroids can be produced in a very short time, higher yield can be produced, and by using biotransformation you can use a higher substrate for conversion. Okay, higher substrate concentration can be used and it will be transformed very easily. So for production of steroids, the starting material you will be using is stigmasterol or diosgenin. Okay, these two are of plant origin. Stigmasterol can be extracted from soybean and diosgenin from root of Mexican barbasco plant. These are used as starting material and these can be converted into different sterol by a combination of chemical reaction and using microbial transformation. For example, stigmasterol is converted into progesterone by several chemical reactions and it is converted by rhizopus to 11-alpha hydroxyprogesterone. Okay, and this can again be converted into hydrocortisone by chemical reactions. This hydrocortisone can be converted to prednisolone, which is used for treatment of different diseases, and this conversion is a biotransformation done by Corynebacterium simplex. Or else hydrocortisone can be converted into cortisone, which is converted to prednisone by Corynebacterium simplex again. Okay, digestin can be converted by chemical reaction into the extreme substrate S, which is converted into the hydrocortisone by the microorganism Curvularia lunata. So now steroids are produced using a combination, and different steroids are produced from the same starting materials. Why? Because steroids are having a similar structure. Then you can also use biotransformation of non-steroid compounds like dihydroxyacetone can be produced from glycerol using Gluconobacter melanogenus. Okay, prostaglandin can be produced, okay, which is used as a contraceptive. It is used for the elevation of pain during childbirth, okay, and for congenital heart failure. So this can be produced from unsaturated fatty acid using the organism Cryptococcus neoformans. Then biotransformation of antibiotics can be done, where you can modify the antibiotics like penicillin G can be modified, okay, into another one, and biotransformation you can reduce the toxicity, enhance the oral absorption, reduce the allergic effect, etc. So you can transform one form of the antibiotic into another form. This can also be done by chemical reactions. Then transformation of pollutants. Biotransformation of various pollutants can be carried out to clean up the polluted environment. And biotransformation of petroleum. There are petroleums are usually in the petroleum producing area. There can be accidental release of the petroleum and leaks, and the soil, the water, and whatever is around the environment is polluted. And this petroleum is not very good for the health. It is a neurotoxin, it is a carcinogen, various petroleum products. Okay, it can cause breathing disorders, etc. So normally mechanical and chemical methods are used to remove this oil spills. But now bioremediation by using microorganisms have been used to detoxify the pollutant and convert the petroleum byproduct to a environment friendly products by the biotransformation reaction. So that is all in case of biotransformation.